This video lecture will focus on the structure of various genomes. A genome is the genetic material of a cell, organism, organelle, or a virus, and its sequence is the order of bases along the DNA, or in some viruses, RNA. Scientists are now able to determine the complete genome sequence for thousands of species, including humans. This information has allowed for the development of fields such as personalized medicine. In this lecture, we will be looking at how genome structures vary between organisms. A genome sequence is merely a long list of A's, T's, G's, and C's, units that represent the order in which the nucleotides occur. Within this sequence are many different types of sequences, including protein-coding regions, those regions that are transcribed into messenger RNA, non-coding regions, and regions that are transcribed into RNA but never translated into protein, such as genes for transfer RNAs and ribosomal RNAs. Interestingly, there is no correlation between the number of genes in a genome, or the size of the genome, and the complexity of an organism. In fact, humans have only a few thousand more genes than those found in a nematode worm. And a simple amoeba has 216 times more DNA in their genome than humans do. No matter the size of the genome or the complexity of the organism, all cells must package their genome in order to fit it into the cell. For example, if we took all the chromosomes from a single nuclei of your cell and put them end to end, they would stretch to over 6 feet long. Clearly, they must be compressed to fit inside the cell itself. In prokaryotic cells, the majority of genomes are circular, and the DNA is packaged into a region of the cytoplasm called the nucleoid. The nucleoid region is composed of DNA that has to be formed into supercoils, or tight, over-twisted regions of DNA, similar to what occurs with a string if you hold it at both ends and continue to twist one of them, as seen here. The supercoiled DNA forms large loops that are held together by a protein core region. These proteins help to keep the supercoiled DNA in a compact state. The supercoils must be unwound to allow for DNA replication. In eukaryotes, the genome is often spread over multiple linear pieces of DNA. Unlike prokaryotes, which have no nucleus, eukaryotic cells must have methods to package their DNA into the nucleus of their cell. This is done through a number of steps involving various proteins. Fully packaged DNA and packaging proteins together are called chromatin. As DNA is packaged, it will progressively get shorter and fatter in diameter. An unpackaged DNA helix is approximately 2 nanometers in diameter. The first and most well understood step for eukaryotic DNA packaging occurs when the DNA helix is wrapped around a group of proteins called histone proteins, forming a structure called a nucleosome. We will discuss histone proteins in more detail on the next slide. This level of packaging is often referred to as beads on a string due to the appearance of nucleosomes in an electron microscope image. It is also called a 10 nanometer fiber based on its diameter. The 10 nanometer fiber is then coiled around itself, forming a structure called the 30 nanometer fiber. This structure is then coiled again and again, relying on scaffold proteins to help hold the coils in place. This additional coiling allows the chromatin to fit into the nucleus. This is also what occurs to a greater extent during chromosome condensation in prophase of mitosis and meiosis. It is important to understand the composition and role of nucleosomes because we will discuss these again when we learn about gene regulation in eukaryotes. Each nucleosome consists of 8 histone proteins organized into 2 4-protein structures. Each 4-protein unit consists of histone protein H2A, H2B, H3, and H4. Histone H1 sits on the outside of the nucleosome, helping to hold the wrapped DNA in place. Histone proteins are rich in the amino acids lysine and arginine, whose positive charges attract the negative charges of the phosphates along the backbone of each DNA strand, helping the DNA to wrap around the nucleosome. Despite similarities between bacterial and eukaryotic genome organization, their packaging processes evolved independently and make use of different types of proteins to bind the DNA and form the folded structure of DNA and protein.